Thank you very much, Simon. I will just share my screen. Now, so hopefully you are seeing my slides there. Yeah. All right, very good. Hi everyone, good afternoon, and thank you very much for coming along to this presentation. I'd just like to start off by thanking the Young ISA Committee, Simon, who I've been liaising with in coming up to this, and to Shirin as well, and to all the committee. The Young ISA is a hugely active cohort of the Irish Statistical Association, and it is fantastic as a good work, and thank you very much. This is also a joint event with the British and Irish region of the International Biometric Society. I'm a member of that society, and I was active on their committee over the last couple of years. So in fact, both of these associations, for anyone that maybe isn't a member of either or both of those associations, student membership is free for both of those associations. So if you're joining in today and you're not part of either of those associations, or if you're part of one and not part of the other, please do feel free to join either the Young ISA via the ISA first of all, or the British and Irish region of the International Biometric Society. Always looking for new members, the societies, so please do join in. So the work I'm going to show today, the title is Statistical Modelling of Ecological Principles and can they be used to advance sustainable agriculture. And this work is in collaboration with a lot of different people. I'm not going to list off a lot of different people, but there's many people involved in various aspects of this work, including my research team here at Trinity College Dublin, and many colleagues from some institutions around Ireland and international institutions. And so I just want to start off by kind of thinking, well, you know, we think about ecology and we think about statistics. So what is ecology and what is statistics? So most of us are from a statistics background here. So ecology is all about the study of life, the study of organisms or plants or species or animals or humans, and how they interact with their environment. So this might be maybe not quite a typical day in the office for an ecologist, but I've taken this image from a group of ecologists that are working here at Trinity College Dublin over in the School of Natural Sciences. So as you can see, quite an interesting day at the office there. Then on the other hand, you have me sitting at my computer. This is a little bit of a different day at the office for me, but ecologists do a lot of varied work and a lot of very interesting work as well. So when it comes to kind of the merger of ecology and statistics, whether you call it statistical ecology or ecological statistics, the merger of the two, some might say, is a match made in heaven. For a few different reasons. I mean, one being that, you know, as a statistician, you get to live vicariously through all of the very interesting day-to-day activities that an ecologist might do, but also for other reasons as well. Ecologists are very much about collecting data, and it's a very data intensive discipline, and so there's a lot of data, there's a lot of statistical challenges, so really it is kind of a match made in heaven in some ways to bring together ecology and statistics. It's a bit of a throwback here to Game of Thrones, if there's any Game of Thrones fans out there. I mean, I don't think this on screen, I don't think this was quite a match made in heaven. I don't think things worked out too well, but I believe things are going better off screen for this. So I do, yeah. So hopefully the idea of statistics and ecology coming together, though, there's many reasons why it's a good idea and many reasons why it works out. So for quite a number of years since I was doing my PhD, I've been working with a lot of ecological data, broader than ecology as well, sometimes in kind of more agronomic or more biological studies. But the idea of bringing together statistics and ecology has been a common strand throughout my career, my research career to date. So what I'm going to focus on today is primarily looking at agricultural production systems and the idea of using ecology within agricultural production systems. So there's agricultural production systems are everywhere. There's all sorts of different systems all over the world. Of course, it's how we feed our human population. There are many challenges when it comes to agricultural production systems, and some of these challenges are to do with populations are growing, so we have an ever-growing population to feed, and so that's a challenge that faces agricultural production. And there are land use pressures. So there's a limit. Land is a finite resource, and if you take land away from one use, such as, you know, if it's an area that's being used as a carbon sink and then you use it for intensive agriculture, that can be a problem. And you know, there's only so much space going around. So there are many land use pressures. There are also challenges when it comes to the climate crisis for agriculture, and this is kind of a two-pronged challenge, because On one hand, you have agricultural production systems being stressed by the climate crisis, by extreme weather events, by very higher variability. So there's a lot of, the production systems are facing drought, they're facing intensive rainfall periods or flooding. So the systems themselves are being stressed by climate change, and this presents lots of challenges. There's the other side as well, in that agricultural systems can actually contribute to the climate crisis. And this is from the emissions from whatever system is being produced, or whatever product is being produced, and that may be from animals, or it may be from, just for example, high applications of synthetic or chemical fertilizer, whereby there are emissions coming out. So in fertilizers, and there are minerals and chemicals that are needed for production systems to grow and to grow effectively. Some of that can be done
Sorry, Caroline.
through, yeah.
It's not in presentation mode, just noticed.
Okay, it's not in
Alright.
presentation mode. Okay, thank you for letting me know. Let me switch that.
Created full screen.
So give me a moment. No. Let me just stop sharing. Okay, is that better?
Yeah, got it.
Okay, excellent. Thanks for letting me know, Simon. Okay, so the challenges then, let me go back into that. So the challenges then, the idea of synthetic fertilizer being applied onto these production systems, there can be kind of emissions problems with synthetic fertilizer, or there can be leaching down into the groundwater. So there's two sides to this. There's the contributing to the climate crisis, but there's also being affected by the climate crisis on the other hand. So a lot of my work over the last while has been focused on developing solutions that can be used to help address these challenges, and the idea of using kind of ecological principles or activity that's going on in ecology to identify kind of nature-based solutions to help to improve the sustainability of agricultural production systems. So I'm going to show you some examples of the work that we've been doing, kind of both from the ecological perspective and from the statistical perspective today. So just to start off, the biodiversity and ecosystem function relationship is kind of an area within ecology. So biodiversity, explanatory enough, is kind of the quantification of what species are in a system, or how they're distributed within the system. And an ecosystem function is the term that ecologists use for a response. So for example, for a production system, that will be something like the yield perhaps of the system. So when it comes to something like a crop rotation system, whereby you have a farmer who's growing some crop, so you can see here kind of like a wheat example, and so you have the idea of, you might have a field where there's wheat been sown, and it might be sown again and again, year after year. So then you have kind of like a monocropping system. And the idea of bringing species diversity into that system can be something that can contribute to the overall system. So if, for example, you think about taking one part of that system and that crop rotation and maybe changing it up and bringing in species diversity, that's one way that you can increase the diversity of a system, by taking one component of it over time and adding in different species. And that allows maybe the species to work together differently, to use the soil differently, and to perhaps improve the soil health, for example, of the overall system. The one way that that can be done is through adding in species that are, for example, legume species that are able to actually fix atmospheric nitrogen from the atmosphere and bring it into the system in a more natural way than applying synthetic fertilizer. I mean, at the moment, synthetic fertilizer is manufactured in plants where you burn oil or whatever to take that nitrogen from the atmosphere and package it up into pellets and distribute it around the world where it's used. And it's very effective for increasing the yield. But systems, so species like legume species, they have kind of these nodules on their roots where they can actually symbiotically and bring in the nitrogen from the atmosphere into the system without the need for that, or at least reducing the need for the chemical systems. So you can have kind of the idea of diversity being brought in across kind of over time within a crop rotation, or even within one part of the rotation, you can look at the species diversity within one part of the rotation. So for example, you might take one part of a wheat kind of rotation and add in kind of a grazed system for a while, where you might have some grassland species, agronomic grassland species now, and that could be whereby you maybe just have one species and you apply lots of fertilizer with it. Or you have the idea that you could bring in lots of species, and you might include some of those legume type species in there. And that will either reduce, potentially displace, but at least reduce the need for synthetic fertilizer. So when it comes to manipulating the species diversity of a system, and in these kinds of ecosystems, they're managed, so you can choose kind of what to sow in your system as opposed to more naturally occurring ecosystems. So you can control what you're putting into the system, and you can then measure what's coming out of the system. So is there a change to the yield? Is it, is this species diversity helping to resist invasion, or are there other benefits to the system as a whole by increasing the species diversity in various different ways? So just to start thinking about the kind of data that you would get from these types of studies. So this is an example from a grassland biodiversity study that was done in Norway. And what you can see here on the x-axis is just the number of species sown, which ecologists call richness. That's one of the fun parts of doing interdisciplinary collaborations. You get to learn the terminology of other disciplines, and you have to start to get the hang of that over time. I'm sure you've all come across that in various different ways. So this here that we've got richness, or the number of sown species, on the x-axis, and the annual yield of the system then over the course of a growing season is on the y-axis. So if you look at that straight away, I mean, if you were to try and just at a very quick glance at that graph, you might kind of say, right, look, this looks like there's an increasing relationship. So go back to the farmer and tell the farmer to sow more species. That's what they should do. And that might be sometimes a good outcome. There's actually a lot more going on with this data. So I'll just tell you a little bit more about the experimental design. So there were 60 plots in this experiment, and there were two different treatment manipulations. So one was the rate of nitrogen fertilizer, the synthetic fertilizer application. So there was like a regular level, and then there was a lower level. And across the plots then, within each plot, the species diversity was also varied. So sometimes there was just kind of one species sown, sometimes there was two, all the way up to 7 species being sown. So there's quite a lot going on underneath the hood there. When you just look at that scatter plot, it's kind of been reduced down to kind of one dimension, but that species diversity, there's actually quite a lot more going on there. So if you measure something like the total yield over the year, you might want to go a little bit more deep than just looking at the number of species and the effect on that. Although this is quite commonly done within this space, just to look at the relationship with the number of species. So here I'm just going to break it down a little bit further. So here in this example, we have the 2 nitrogen levels. So I've just split it up there by the 2 nitrogen levels. And you can see that there's a bit of a bump up, certainly when you go from the low nitrogen level up to the regular nitrogen level. But then all sorts of other questions like, well, is this nitrogen effect, is it consistent across the varying species diversity or not? And in this case here, there were 2 grass species and 2 legume species within the 7. And so the questions will be, well, how do those species work together? Can they interact synergistically? How does the nitrogen fertilizer impact? Does it impact it in a species individual level, or what? So here's just another visualization where we're getting to grips a little bit more with what's actually going on at each of those data points. So you can see here both the difference between the low and the regular, but you can also start to see what's happening across the species diversity gradient. So the grass species have been colored here within each pie glyph as a blue species, ranging in shade. And then the orange and the red 2 colors there, the 2 legume species. So even at a glance, you can kind of start to pick up some patterns as to what exactly is going on within the yield versus the number of species. And you can see that there are certainly differences on account of maybe the identity of the species in the system. So how do we go about modeling this type of output? So the data modeling approach that we've been using here over the last number of years is called diversity interactions modeling, and it allows us to kind of break down, not just kind of looking at kind of like a univariate kind of metric or some sort of univariate measure of species diversity, but to break it down into, well, how are the individual species contributing? How are the species interacting with each other? And then on top of that, how are any additional treatments going or affecting things? So there's a few things going on here. I'll just explain. So the first thing, the P's here are the individual sown proportions. So you have there the sown proportion of a species in an individual plot. And there's an associated term with that. So we have a beta for each of the individual species. We then have pairwise interactions. So how are the pairs of species working together? Like, are there some pairs of species that will actually get on really, really well together and work synergistically and improve the yield a lot? Or are there species that actually shouldn't combine together that maybe have negative interactions? We also have then the term for the high end treatment. And of course, there's lots of different ways that high end could come into it. So this is the framework. This is just kind of a high level view of this framework. There's obviously a lot of different ways that this framework can be used. There's a lot of different ways maybe that species might interact, and some of that might be biologically motivated. And of course, there's a lot of different ways to frame how any additional treatments would enter into the model. So just kind of a quick kind of aside here on this, like, so I've showed you kind of an example from 7 species, and of course trying to think about a predictor space that has dimension n equals 7 is a little bit tricky. So just very quickly, if we just kind of think about this modeling approach from like a 3 species example, like effectively what we're doing is kind of saying, right, well, we have this kind of compositional data, we have these proportions that are our predictor space, and that predictor space is really comprised, like it's making up a simplex space whereby, of course, at each point those proportions have to sum to 1. So really what we're doing is fitting kind of like a response surface type model where you're looking at, well, how do the individual species affect the response? And here the individual species are shown at kind of the ternary the vertices of this ternary diagram, and then how are species interacting. So in this kind of just quick hypothetical example, you have the situation that species 1 is performing higher than species 2 or 3 when it's on its own, but when you mix it with species 2 and maybe a little bit of species 3, you get synergistic interaction that will actually do better than species 1. So of course, when you have dimensions equal 7, that becomes a little bit trickier to envisage, but you get the idea that we're dealing with kind of a continuous surface space here for our predictor space. So just going back then to the Norway data, so this was the final model for the Norway data. So I'll just break it down a small little bit. So we still had the term for each of the individual species. So this will look at how each individual species contributes to a response. We also have interactions rather than an individual pairwise interaction. There were just 3 interaction terms in the model. So the first one here is a term to capture how any grass will interact with any legume. So it's kind of being driven a little bit by a biological motivation that we know a little bit about these grass species and these legume species. So it'll just capture how any grass will interact with any legume. We then have this term here will capture how any grass will interact with any other grass. And then the last term there will be for the 2 legume species, how are they interacting? And so the last bit there then is about the interactions. So there's quite a lot of model selection went on before this final model was selected. But the interaction there, we've got the additive effect of the nitrogen treatment, but also the interaction between the nitrogen treatment and the proportion of sown legumes. So PL there is the sum of the 2 legume species rather than individual ones. So this is telling us that there are constant effects of nitrogen, but that they're also affected by the sown proportions of the plants and whether or not there's legumes in that. So just a quick look at how this model will look. So we end up, in this case here, we end up with 7 terms for the individual species. We have the 3 terms for the interactions, and then we have the 2 terms that are capturing how the nitrogen treatment is working out. And so just a quick look at what's going on here. This is a predicted response. So you can see straight away that there is this increasing relationship with richness, but there's also this kind of saturation. And if you kind of look a little bit closer, like the individual points here, they're not kind of replicates of the same thing. They're not just showing variation across the same type of replicate. The composition is actually changing within each of these points. So when you look at richness equals 3 and richness equals 4, you can identify combinations of species that will actually do better than the higher richness species or the higher diversity. And the same at the end level, there's a little bit of a bump up, but we can kind of find combinations of species that will work very well together. So just to pick out a few of those communities, just to kind of show you a little bit more, you can see here, what you can see here is the predictions for each monoculture of species. So the grasses, there's a constant kind of shift up. For the legumes, there's no actual benefit to adding nitrogen to a legume monoculture. And this kind of makes sense quite biologically. But when it comes to the different mixtures, I've just pulled out 3 different mixtures there. One where there's grasses and legumes, and then the last one where there's the 7 species mixture. And you can see again that there are communities that can do just as well as the 7 species mixture. So I guess the take-home message here is really that the species identity does absolutely matter, but it's important to mix the grasses and the legumes. That came out very strongly, but also that the identity of the grasses is actually more important than the number of species, or the number of grasses. So for example, the first 3 grasses are performing better than the last 2 grasses. And so it's not just about increasing the number of species, it's also about looking at more kind of nuanced ways of combining these species. So there are optimization methods whereby you can look at, well, how can you identify kind of the optimal performance? And that might not be one community, it might be a range of communities. But there are optimization methods that we can use to identify that. So just to kind of capture, maybe to kind of so the example I've given you there is kind of a single site, a single year, and 7 species. So I'm not going to go through all of these statistical challenges, but I just want to give you a flavor for the types of challenges that come up with this type of data and the types of challenges that I've been working on over the last number of years and continue to work on in some of this space. So the first one is that you can, of course, have maybe many species. So the example I showed you here, I gave you a hypothetical example where we have 3 species. And of course, if you have 3 species, there's just going to be 3 pairwise interactions. The Norway example, there were 7 species, so that would be 21 pairwise interactions. And then for, say, if you have 15 species, you would then have 105 pairwise interactions. So straight away, you can kind of see that as the species are increasing in number, the number of pairwise interactions and the potential complexities gets a lot greater. So if you just even think about a system where you've got 50 species, and there are, of course, systems like that. There was one system I was working with where we had 72 species in total. So it is feasible that you would have that number of species in these types of ecosystems, maybe not in agronomic systems, but in other types of systems. So here you'd have over 1,000 pairwise interactions. So straight away, you can see like trying to find biological meaning in that will be quite tricky. So the type of ways that we have done that is, first of all, to look at those individual pairwise interactions and find ways to kind of exploit that. That might be based on the biological properties of the individual species in the system. So the Norway example, what we did was instead of having 21 pairwise interactions, we broke it down into 3. And this would have been, via a model selection process. It wouldn't have just kind of been by choice, but that idea of kind of grouping species that have similar traits came from the biological motivation, and then it was statistically tested through the model selection process. So what we've done is this idea of exploiting the fixed effects, but also then recognizing that there could be a lot more variation going on that maybe wouldn't be captured by those fixed effects. So what you can see here in this equation is the second equation here is the first component is again the identity effects. So looking at the species specific contributions. Then we have the interaction terms that here I've just characterized it by a single interaction. And then the last part there is actually a random effect where you're saying, right, well, there is additional variation. So technically what we've actually done here is added in 1,225 random effects, that's the delta IJs, but have forced them all to have the same variance. So effectively what that's doing is just adding in one extra parameter, and it's recognizing that, okay, you've captured the interactions in some more parsimonious way, but you're recognizing that there is variation at the individual pairwise interaction level, and that additional parameter can capture it, and it can then feed into the inference for the model so that that isn't just assumed to be random error. So a major difference here is that, typically when you think about random effects, they're usually indexed by something like a block or maybe like a person effect. But here the random effects are actually indexed by the species within the plot, which is quite a difference to the typical kind of mixed model that you would see. But it's one way that we have captured kind of the complexity of the system, but to reduce it down so that we can still find biologically meaningful outcomes when it comes to looking at how those species interact. So just to pick up on one or two quickly other challenges that we faced. So I mentioned already about multiple kind of responses, and the example I showed you, I focused in on yield. And yield, of course, in an agricultural production system is very important, and it's often hugely of interest. But of course, there's lots of other things that are of interest, and you don't want to maximize your yield and then find that you've developed a system that's very, that might be very susceptible to disease invasion, for example. So the idea of looking at multiple functions is very common nowadays, and this idea of how can you kind of capture trade-offs. So you may have an example whereby certain parts of your species compositions will allow you to promote maybe multiple functions, but maybe not all of them. So we've been developing methods to look at these multivariate responses. So you then have kind of multiple dimensions, both on the response side, but also on the species diversity side, and coming up with kind of sensible ways to capture that make sense statistically, but also that you can find biological meaning can be a little bit challenging. But so we've been doing some work in that space. So here's just an example of one of those whereby what we've done is kind of looked at kind of on average how various different compositions are looking, also looking at how they are affected by different stresses. And this example here was where there was kind of a drought was implemented on these individual grassland species, where you can see here what you're looking at is kind of an overall kind of average response across several responses, but also you're looking at the individual responses as well, so that you can capture that variability within a particular type of community. How is it performing across multiple responses, but also are there trade-offs? Like, are there some communities that will do really well? So the wider kind of ranges here are communities that are very mixed across the responses that we studied, and the ones where you can see kind of the various responses are more clustered closer to the average is where it's maybe performing consistently, and it can be performing consistently poorly or consistently well. So that's one of the areas that we've been developing as well. And of course, then with any kind of ecological study, the idea of how do spatial patterns affect any of these ecological processes is often crucially important. And so many of these studies, you might have data collected from multiple sites. So in fact, the example I showed you did actually have several sites in that study across Norway. So one way to deal with that is to develop these models within a mixed modeling framework, whereby you either add in kind of some way to vary things from site to site, or what I'm showing you here is actually a random coefficients model. So if you have kind of like a random intercept, you're saying that each site kind of shifts up and down. But if you have a random coefficients model, what you're actually allowing is that the individual terms in the model can vary across sites. So for example, you know, here you can see in Norway, like there's a huge gradient, and you have the site up the north, up in Tromsø, which has very, very severe weather and would often be covered in snow. And it might be that some species will perform very poorly at a site like that, but others might flourish. Whereas if you have something further south, many of the species might do well. And so to be able to allow those individual species effects or how they interact to vary across sites can be quite helpful. So of course, then those kind of mixed models, they come with lots of challenges, because again, you're adding in these additional dimensions. So of course, yeah, it comes with additional challenges and additional overheads. So just maybe to start to wrap up, these challenges, you can look at all of these challenges individually, but you can also have the situation where many of them, or perhaps even all of them, can occur in the same data set. And of course that does happen. And then you've not just got kind of individual challenges to deal with, but you have to think about how to jointly deal with these as well. So I'm just going to finish up with one example of a study that I'm working on at the moment, which is a study called LegacyNet. The website address is down the bottom there. So this is an experiment that has been conducted across 26 different sites over the world. And the experiment has been going on for the last number of years. So I've been deeply involved in the setting up and the running of this experiment. And so the data has just finished, the experiment has just finished at all sites by the end of last year. And so we have now got the database from across the experiment being collated and being analysed. So the statisticians are really coming to life now in this project. But this is looking at this, just bringing it back to the crop rotations that I mentioned at the beginning. This study is looking at crop rotations. So what happens if you manipulate species diversity in one part of a rotation? Can you have maybe, say, legacy benefits on another part of the rotation? So we've been examining how changing species diversity in one part affects the system as a whole. And of course, what you have then is many of the challenges that I've listed here. We've got we've got space, we've got time, we have the species proportions that you sow will change over time. So how do you build that in dynamically if you want to look at legacy effects? And of course, with this kind of scale, you end up with lots of estimation challenges, which of course are fun to deal with. So that's just kind of an example of a lot of the current work that's ongoing with my group and collaborators. So just finally, just this modelling approach, there is a website, Diversity Interactions Modelling, if anybody's interested in finding out more. And we have been developing OR packages for using this approach for statisticians and also for ecologists. So we have both kind of modelling packages, the first two that are listed there, but also a visualization package to help with dealing with the dimensionality challenges with this type of data. So finally, thank you very much for your attention, and I will just finish up by saying that there are lots of interesting challenges and topics in ecological statistics. It's certainly an area where I have found that there's been a lot of engagement, and there's a lot of very quantitative people working in ecology, so it can be an excellent way for statisticians to have meaningful collaborations. So certainly from my experience, I would totally recommend it. So thank you very much, everyone.
Cool. Great, thanks. It was really informative, really interesting, and yeah, just really cool to actually see like such large scale experiments. So yeah, we can start with some short questions. So yeah, I guess you can raise your hands or just post in the chat. I have a question which is probably kind of a bit more on the interface of the data versus what actually happens on the ground. How accurate are the proportion measurements in the first place, and how homogeneous are those proportions kind of throughout the plot?
Yeah, that's a very good question. So there are ways that you would establish and decide how to kind of combine the seed mix. So to
Mhm.
do that, you would kind of do things like, well, what would be the typical seeding rate at that site? So how would that species perform at that individual site already? So it's not completely trivial to even combine the seeds to sow it. The seeds then are typically mixed up so that you would expect in these types of systems that you would have a space, you would have kind of random kind of uniformity across the plot. Now I have looked at studies where the spatial variation within a plot has actually been manipulated, which is a whole other level of spatial
Okay, yeah.
variation. So you can do that, but for the examples I showed here, you would aim for a uniform kind of across the plots. So when it comes to the establishment of the plot, then you would assess that quite early on, because you can have a failed establishment for things like, the seeds didn't work out, or there was just really bad conditions the day it was sown. So you would monitor that early on, but providing that there is a good representation early on in terms of what you had intended to sow, then any changes after that are to do with competition of the species, and they're to do with kind of part of the outcome of the experiment. So then you would kind of be tracking it for a different reason. There are different techniques to capture those proportions. Some of it is done visually, and there will be a lot of training for individuals, and it would usually be performed by the same individual to ensure consistency, so that at least within a site you would have relative consistency. And you also have it's like grab samples, whereby you will be doing kind of manual separation. And there's been a little bit of work done on kind of things like, well, at what stage does the variance of the proportions start to reduce? So based on the number of samples. So the more grab samples you take, then over time, the lower the
Gotcha.
variability. But I mean, I had this conversation early on in my career with somebody, and I said, oh well, you could just kind of maybe just separate all the plants and see how they compare to your grab samples. And they roared laughing at me. They were like, look at the size of my field. And I did some separations for a day once or twice, and I tell you, like, it's tough work. Like, so I would say there's absolutely variation associated with it, but it is
Yeah.
quite consistent.
Okay, cool. Awesome. I think Nastron had raised hand. Nastron, have you a question?
Yes, sorry. You mentioned some challenges in estimation, and I think the most challenge can relate it to add random effects to your model. I like to learn more about this now. For example, how did you overcome on these challenges by after adding the random effects in their estimation process, and which techniques could be helpful to overcome on these challenges?
Yeah, you've hit the nail on the head there. That is exactly part of where this challenge comes in, because as soon as you start adding in random effects, and particularly the random coefficients models that I showed, where you might have 10, 15 predictors that are then all being allowed to vary randomly across sites. So you can have a variance covariance matrix there that's quite large with quite a lot of parameters in it. So typically what we do is we simplify things down, first of all. If we have, if you go to the optimal, where you allow everything to vary freely, then you're likely to hit convergence issues. So I generally follow a procedure of kind of break things down, make things very simple, and see where things will start to work, and then start to build up the complexity. And quite often you don't need to go to the case of having an individual variance parameter for everything and an individual covariance for everything. And this is where some of the discipline-specific knowledge, like the ecological knowledge in the collaborators, can come in, because there sometimes are biologically meaningful ways to kind of build in covariances, like say, okay, well, these 2 species perform similarly, or these 5 species maybe perform similarly, so let's allow them to co-vary in the same way. So there are ways to simplify things, but yeah, it's a major challenge, absolutely. So thanks for that question.
I think, yeah, to kind of keep things on track, finish it here. And yeah, if you have any kind of more general questions or more specific questions for Caroline, you can post them in the chat or message any of the committee members.